Thank you very much. A question for Dr. Atocha. Amazing talk, and I'm always quite amazed to see that the same bacteria are coming up again and again in non-small cell lung cancer, Akkermansia specifically. And it's one of our top bacteria in North America, and now in Spain too. This suggests that this might be independent of diet. We can imagine that patients in Spain eat very differently than patients in North America. Do you collect the dietary history of these patients? And maybe is this something that you plan to do in future FMT trials?
Yeah, this is something we plan to do in FMT trial, but we did not include that in the easier in NADIM 2. Yeah, but it's true that yeah, that is important information that we should take into account. We have yeah, body mass index, but this is the much information that we have regarding diet of the patients.
Sorry, also a microbiome wannabe. One or two other questions there. There's, expanding data in that microbiome space that PPI, fiber intake, and maybe steroids might impact these associations. Did you collect that data and control for it in the analyses?
Yeah, we have that data, but we haven't, so roughly, we haven't seen anything, but we need to look more deeply. But we have the data, and of course, for the publication, we will report the data.
So it's really just a comment I have, and it's probably directed to the first speaker. That abstract that you talked about that made an oral at ASCO looking at the circadian changes has gotten a lot of press. And I'm not a believer. I'm not drinking this Kool-Aid. Whenever you have a trial that size, you have to ask the question, what is the science before it? First of all, they looked at only 4 cycles of Pembro. And the second thing is that doing the treatment before 3 o'clock makes no sense. When you go into their paper, they looked at all the previous trials that sort of showed the signal, and they picked 3 o'clock, but that makes no sense. In the same way that you have to have your surgery done before 4 o'clock, that makes no sense. So whenever you have a treatment, a study, you have to have a scientific, biological reason for it. The senior author that they looked to, besides Tony Mock, was from France, and in his paper, it was actually 11 o'clock. That senior author has well been talking about this for 20 years. I used to treat GI malignancies as well, and he tried to prove to us that we should get our chemo and our radiation before 11 o'clock. So I know it sounds really easy and it seems really simple, but people should not be getting on that bandwagon so quickly. And I'm probably the only medical oncologist you meet that doesn't actually believe this.
Thank you for the clarification. I'm just a radiation doctor.
I don't know if I'm supposed to interject, but can I put one of my colleagues on the spot to maybe address that comment? Dr. Spicer?
Yeah, I think there is actually quite a bit of biological rationale behind that work. There's tremendous variability both in blood and tissue on a diurnal basis in terms of all of our immune lineages, and that's been proven in a number of settings, and we have our own evidence supporting that from our own work. So I think it's in a randomized design, it's a little bit bold to dismiss it out of hand. I think there actually is quite a bit of science behind it in terms of its applicability and how we translate it. I think that's maybe another matter, but I just want to say I absolutely loved your talk. It was additive to Dr. Jabor's work, and it brought a lot of insights that I hadn't previously appreciated about radiation. So that was thank you for doing that. Maybe a question for Dr. Zhang. Again, I don't know if you guys appreciate this, but he's quite the biologist, but also a technical champion and innovating all this stuff. To what extent are you guys relying or letting the artificial intelligence guide your decision-making in the tumor board? Do you find that there are moments where the AI decision generated is different than what your tumor board would have recommended, or how do you resolve those kinds of discrepancies?
Yeah, from this year's Spring Festival, in China, that there's a DeepSeek. Do you know that? Just like the ChatGPT and it's deep thinking, but the DeepSeek is open. The algorithm and the software is open, but it's on public, and we focus on the lung cancer disease and establish AI treatment strategy, MDT. Now, in the outpatient, we will rely on this system because he can organize the data very high efficiency. And because some patients come from the other hospital, there may be 100, including the pathology diagnosis and genetic profile and so on. We scan it into the PDF, and he will organize in very short and very precise, and we can make a quick, very high efficiency. And we found that and we will input our own center's case in the system and make it more reasonable. Because too many in the DeepSeek system now, just like the Baidu, and many rubbish and many advertisement information, it make it not very evidence-based, and we will make it more evidence-based. And I think it's very clever, and he combine the evidence-based medicine and doctors' experience and the patients' design together.
So, question to Dr. Romero. From your talk, I got quite confused because there's a lot of stuff in there.
Yeah.
And I kind of understood that your focus is to try to correlate the microbiome with potential metabolic signatures, or is that two different components? So if it's the same, track, so do you think that the metabolic activity of the microbiome is the determinant of cancer activity more than the immune effects of having bacteria in the gut, or maybe both?
Well, it's not, so the objectives of the study is just to describe what I've seen. So regarding metabolics, we plan to do a more targeted approach and validate the metabolomics that we have been found using the data with the real Nadim, because we have been analyzing many metabolites, and the detection was semi-quantitative or quantitative, but very roughly, but we want now to really select those metabolites that have a signal and then validate them in the 100 cohort from real Nadim. So we don't have a previous hypothesis of what is more important, the metabolites, or the idea is just to describe. And but yes, one of the things that we want to do once we have validated these results is to identify or correlate these metabolites in plasma with microbiome with the gut microbiome, and see and try to understand what is the mechanism by which microbiome shapes or modulates immune response. We will also, but we don't have now, data from spatial transcriptomics. We will have data for different cells of the microenvironments, and that will also help us to, maybe understand what's going on.
So I may embarrass myself in front of my microbiome colleagues, but is there any value to look at metabolites in the stool to make that frontier between plasma and microbiome?
There are some people that is doing metabolites in the stool, but we didn't that. We measured the metabolites in the plasma sample, so we didn't look at metabolites in the stool, but there are some groups that are also looking at metabolites in the stool. But yeah.
Are there any other questions or comments? If not, I'm going to take advantage of my soapbox here and ask a quick question / comment for Dr. Willer. So, if you could comment on the timing of radiation therapy and the adjudication of immunotherapy. This is sort of what you see in the radiation literature with the combination of the two is kind of in stark contrast to what you see with the neoadjuvant trials in surgery. So surgery is sort of a terminal event. You just take it out. It's a bit dumb. We don't really know what's going on. But radiation, I would imagine, has different phases of effects, right? You would imagine acute and then a more prolonged phase, an acute inflammatory versus a more adaptive phase. And there's a lot of evidence in the basic literature to suggest that acute inflammatory responses or myeloid responses and associated with lymphopenia are very suppressive and associated with adverse outcomes. I just wondered, do you think the timing of giving your immunotherapy, are you like kind of quenching the effect of your radiation therapy? Like that's I guess the gist of my question. If you could comment on that. You're welcome. I'm sorry.
It's a good question, and I only have hand waving for you. I think by and large the timing of radiation should not really matter. But I mean, again, as the ASCO data suggested, there's a lot more that we do not understand, and radiation has turned out to be more complex than we think. And it is very well possible that if we don't get it right, we do not get the synergy that is there. There's no question that radiation is immune stimulating, although for some reason my surgical friends like me more when I say that radiation is immunosuppressive. I don't know why that is. But, we have to get the balance right, and there's a lot more research that needs to be done.
Thanks. I would imagine it's a bit of both, So, anyway, thank you, everybody. I guess we're right on time.
Yeah.
So with that, again, thank you guys. It was great talks.
Thank you.
Fun.